Welcome to Neurosalience, the OHBM podcast.
Welcome to the Organization for Human Brain Mapping Neurosalience podcast. I'm your host, Peter Bannattini. Here I explore advances, challenges, controversies, and opportunities in brain imaging and modeling with neuroscientists around the world. So today I have the honor of talking to an old friend of mine, Dr. Vesa Kiviniemi, who is a radiologist and researcher in Oulu University in Finland. We were talking to him during a massive snowstorm going on in Finland. So he had all kinds of time to talk because he wasn't going anywhere after that. So Dr. Kiviniemi received his MD in 1996. He served as a radiologist. He also has a PhD as well. They do both for this program in Finland. After a stint at Helsinki University as a scientific fellow, he spent 3 years at the Medical College of Wisconsin, working with Bharat Biswal and Andrej Zmanowicz and all of the people there, learning about resting state fMRI. And he returned in 2003 in Oulu University, or maybe 2001 in Oulu University, and he's been there ever since. And he has a great group, and his focus recently has been on using an extremely high-speed MRI technique called MREG. It was developed some years before, I believe, by Jürgen Hennig's group, and it basically allows you to collect an entire volume of data with a TR of 100 milliseconds, and it just uses a stack of spirals technique. And the reason why he likes this technique so much is that he can actually look at all these different types of brain pulsations, cardiac, the respiratory, and what he looks at as glymphatic or CSF pulsations. They're all modulated in different ways, and he doesn't have to worry about aliased high-frequency information, at least most of it, from the heart, for instance, that might contaminate some of the signals. So I think it's a great technique. I think it's a great sequence. More people should be using it. And he talks all about how he's used it. And also we talk a little bit about the history of our understanding of the glymphatic system and what we think it might be doing, all the unknowns about it, potential opportunities of research, and among other things, why using this high-speed technique is one approach that might be complementary or even better in some ways than approaches that are slower, that might be more specific to things like CSF or other aspects. We're still struggling to image it well, image the glymphatic system function well, but we're so far, I think that what we have obtained is really interesting and really useful, and he's, as he's talked about, potentially clinically already very relevant. So it was a great conversation. He's a lot of fun to talk to. He's very open and easygoing and really knows what he's doing with regard to this line of research and so many other things. So with that, I hope you enjoy the conversation. It's an honor to be here with Vesa Kiviniemi, who is a professor at the University of Oulu in the middle of Finland. And right now they're experiencing a snowstorm.
Oh, yeah.
And today we're going to talk all about his work primarily on exploring the glymphatic system with MRI and specifically with MREG. But we're going to talk about a few other things too. And basically the glymphatic system is something that's sort of, we'll talk a little bit about the history, I guess. But it's really caught on, and a lot of people are really trying to look at it with high fidelity, and because it seems like it brings together a lot of aspects of brain health, brain function. It seems to be something that's worth studying for understanding the brain and clinically. So welcome, Vesa.
Hey, thanks for the invitation. This is great, and this is gonna be fun. I have really been looking forward to this night, and now it's nice that it's bad weather outside. I can stay all night long. No problem.
All right. Yeah, no, it's actually, and I've been looking forward to talking to you about this as well. I mean, this is something that I've talked to a few other people about this, but your approach on this, I think, at least you have the most papers and the biggest body of work on looking at humans, and yeah, and I think that's I'd be really interested to see what you've done, and also, of course, I've known you for years since.
Oh yeah.
You've been at Medical College of Wisconsin. You've spent a brief time there, and
Yeah,
well,
yeah.
Let's just start off
Okay.
by just talking a little bit. I usually like to start these podcasts just by talking about the person's history. It seems that you have you have an MD, but do you have a PhD as well, or
That's what we get here. So as I get an MD, PhD from our system, and but I'm a medical doctor by profession, and I'm a neuroradiologist by specialization. So that's my angle to it. So yeah, and I was lucky enough to get also to MCW like you were. So I'm honored to be in your podcast. This is MCW guys talking. So it's in a way, if I'm allowed to say that, I would like to see it that way from my point of view. So that's
yeah.
fantastic. Yes, also in that respect. Yeah.
Yeah, and so you were at MCW at
Yeah.
like early 2000s, I think, right?
Yeah, 1999, 2000, 2001, on and off. Not exactly a year, but something like that. So a little bit less than a year. So yeah, I popped by there, and I remember when I went there. Bharat has this R01, and we got some funding, and I was able to go. And I remember walking to the old Brugger, and then I saw posters on the wall by Pandetini and Wong and Rayo, and I'm like, holy cow! I had no idea I'm getting into this place. And I was like looking, and I went to the Brugger, and Andre was there, Jim and Bob Cox,
Yeah.
and Rasmus, all the guys. So
Yes.
that was fantastic. That was just mind blowing. Oh
Yeah.
holy cow! Yeah.
I'm surprised they still had our posters up. I mean, I had left there. Well, maybe they did have them up then because I had just left around
You
1999.
had just left, yeah. So it wasn't that far off, yes. So yeah. I just met you and Eric. You had all both gone. So, but Ted Dio, Jeff Binder, Elliot was there. So still quite a few guys, but
Yeah, it's 9. Yeah. And, honestly, just as a sidetrack, right? I mean, there was this mass exodus that started actually with Mike Beauchamp. He was a scientist there. He was working with Ted Dio. He left in the early 90s or mid 90s. Then, Bob came after I came, and then
yeah,
then Rasmus was my postdoc, and
exactly, yes, yeah.
but yeah, no, it was a mass exodus. I don't know if Jim ever fully forgave me for that, but oh well.
Well, yeah, it's a heck of a loss, but what can you do? It's NIH is not a bad place to be either.
It was a great place because it was small and you could do many different things that were a little bit outside the box. They had a machine shop. They had a lot of creative people, and it was like very low overhead. And I think that's key to a lot of the things that happened there.
Yeah, and Jim had this environment, from his patent grants. It's just amazing. And after realizing when I went there and came back, the amount of information I got and hands-on shimming on a 3 Tesla there, it's like,
oh yeah,
you learn so much. It's just right on the focus. So, and the Bruker was what it was at then, and then saw people, Andrei Chesmanovich coming with a paper and a black ink and producing these perfect shims for individual brains. You're like, it's still unbeatable stuff. It's Andrei, right? It's like.
Yeah, there were all these knobs that controlled the the various interactions of the shims.
Oh and then they were polynomial, and they gave me one time opportunity to do it, and I completely messed it up. It's
there's no way of having an intuitive feel for that because it's so nonlinear all over the place, and so
it's just like boom boom crazy. So that was nice experience to learn, and they were laughing their ass off behind me. That was nice.
But one thing actually just to mention about the shim, and it's something that the companies today could do, but they don't. like Bruker was nice in the sense that you had shim files that you made for each person's head. And so when a person came in, you could just bring up the shim
Oh,
file because each person's head is shaped a little bit different. And
absolutely, yes.
yeah, and so you could just upload that and boom, you were shimmed. And it
Oh,
was
wow.
it was perfect. And you could do that now, but they don't. So
Oh, yes. Oh, yes. I hear you. I hear you.
yeah.
Yeah. Yeah.
But
Those
that's
were the
another
days,
topic
man.
altogether. Yeah.
Those were the days.
Yeah. So you actually working there, and then afterwards you came back to
Yeah,
Oulu.
yeah. I came back after that. Bharat went to New Jersey, then Rutgers, and then I came back. And so why I got there was I saw Bharat in the Philadelphia ISMRM buy his poster for once, and
Yeah.
so I went to him because I wanted to do a sort of calibration study on anesthetized kids' brains, and you had the story that you have to have these activations to see the ball response, you know. And then I did the anesthetized kids fMRI with our GE 1.5 Tesla echo speed, and I saw these fluctuations on an anesthetized kid. Then I did your FFT analysis, and we had some excellent coding guys there. They made it work in one day. We had our own phantom software like AFNI for
Yeah.
DICOM images, not for a Nifty or anything like that. It was DICOM.
Yeah.
And actually, we got a spin-off firm out of that again nowadays for like clinical, looking at anatomical images for
Yeah.
clinical work. So that's also really nice. So I saw these funny fluctuations, and then I could map visual cortex with that. And I went like, what is this? And then I talked to Bharat, and he went like, oh, really?
Ah,
And then I got to MCW, and we started looking at all sorts of things like anesthesia effects. We tried with Tony's rats, but I was trying to use thiopental and midazolam that we were using here, but
yeah.
that wasn't a good idea for rats.
Yeah.
We didn't know it at then. Then Chris Pavela later on, based on that work, partly went to dexmedetomine, which works like a charm. That's perfect. So
Okay.
it's wonderful work there. So it's an improvement there nowadays. So
Okay.
yeah,
Okay.
that was
Okay.
the thing. Yeah.
Cool. And so your work, you've done a bunch of things. So just before we get into your main thing, I just remember, so just out of nowhere. So I looking at your Google Scholar, and as I do the research for this, I see
Yeah.
you're looking at things like the effects of surgery on primary cortex activation, ADHD, autism, psychosis.
Yeah.
And one thing that caught my eye, and back then, and I still think it's relevant today, is the effects of light shined in the brain.
That's an interesting thing. There was this biologist here who had been doing a lot of animal research, and it is known that there are opsins in the brain, and melanopsin, panopsin, neuropsin, opsins 3, 4, and 5. Opsins 1 and 2 are here in the back of our eyes, and we are using it to see, obviously. But it's a peculiar thing. There are opsins here in the brain, and then like here right now, this is the worst time of year because we don't have much sunlight, and we get this seasonal affective disorder. We call it darkness depression or whatever you might call it.
Yeah.
We have a HPM paper about that the connectivity in the visual and occipital areas increases in these patients compared to those who don't have it.
Okay.
Then there was this guy who wanted to try to develop a treatment just by putting the light directly into the ear, where there's actually, so blood and skin are the ones absorbing light, but not the eardrum nor the bone. So you can get a lot of light into the brain. And we saw some changes, also behavioral changes in our ice hockey team, and we have published those. Also
Really?
shown
Okay.
that there are opsins in the human cadaver, forensic patient brains in slices. So there are opsins in the brain. And Guyton, the physiology book writer, has a paper that during a very bright day you could read a paper inside the human brain. There's so much light entering the brain.
Interesting. I always thought the penetration was about like maybe, obviously with the skull and everything, it's,
Yeah, that's what I thought, but that's not the case. That's what they kind of measured somehow, and then there is this Professor Wong in Singapore, they put light into the visual cortex from the back of the macaque brain or something, some animal brain, and they got the visual system activated. So,
Wow.
yeah.
So it's also my connection to all that is way before I did fMRI. I was an undergraduate and I was working in a pharmacology lab, and this professor Janice Ells at the Medical College of Wisconsin. After I left, long after I left, she took up this thing, and it's similar. It's called photobiomodulation with near-infrared light. It's considered sort of a therapy for traumatic brain injury. You just simply have near-infrared light you shine at your head, and it's not necessarily the opsins, I don't think. It's just simply the mitochondria are somehow light sensitive, and they're more energized apparently. And it slightly increases the metabolic rate.
I haven't heard that. That's a new thing. Yeah, wow. Yeah, I mean,
near
I've
infrared.
seen on and off these near infrared therapy options. Somebody had a helmet for Alzheimer's saying that it helps and that stuff. But it's been going big. But that.
It's a little bit out there, and it's
exactly.
But,
yes. But they get funding and then moved on to something else. But So we have fundings due to our day is it's gonna be 4 hours long daylight. So and if you go further up, there's not gonna be a day at all. So
yes, and that has a real effect on someone.
Like people coming from south really get affected because they're not used to it at all. And I get slower down right now, so I'm not at my prime, unfortunately. So summer is a completely different ballgame. All day long light is like whoa.
Well, it's interesting. I actually remember reading a paper on, so this is totally off track, but on how depression is sort of an evolutionarily, you get depression for a purpose, to hibernate. So like just you don't want to move, you just want to sit there, and you're kind of depressed. Sure, you're depressed, but you're doing what the right thing is to survive when it's cold and dark and whatever. And obviously, And so when it gets light out, you get more dopamine, you start foraging, you start looking around, and then you get happy. And that sort of drives you to forage.
And this is also off the topic, and I don't know how to show it, but I've been thinking that this glymphatic clearance thing might be related, because the light it's a natural free controller of your, like you were saying, when it's dark, you get to go down and the activity goes down. And when it's light up, then so it might be controlling also that. I don't know how, but this is just the idea. It might be somehow because it would be automated and cheap.
Yeah. And so you've been looking at sort of the effects of, with fMRI and other methods, just things like psychosis, but you're a radiologist, but you're also obviously collaborating with people looking at who have psychiatric disorders and
neuropsychiatry, neurology, psychiatry, surgery. I'm a neuroradiologist, so I'm in the serving business of other clinics. So I'm in a nice place to do research because we get all the patients and we just collaborate. And I have really good colleagues here that are really helpful, and it's sometimes small is beautiful, like here. So
yes, I tend to think that just like we were talking about with MCW, I tend to think that a small group that works well together is almost the best sort of unit as far as that's concerned.
If you get a good thing going, it's invaluable. It's not always that easy to come to that situation. Money isn't all. Of course it's important to get things going, obviously. Yes, you need a scanner and stuff like that, but that's the people and how to get them humming on the issue and focusing on something. I've been really lucky with the people now for over a decade, so it's really nice. So
that's great. All right, so why don't we just get into the glymphatic system then? Maybe first start how you got interested into it, and maybe give a little history. Maybe starting with Megan Nedergaard, and also she mentioned somebody who's at the. I remember just looking at her talk. She was mentioning somebody at the NIH who's now just a program officer who had discovered the glymphatic system, but nobody ever took it seriously. And then she just
Rennels. Is it Rennels?
I'm not sure. I'll have to look that up.
There are something
All
like
right. Well, I'll look it up, and as we go along here. But yeah. But either way, Nidegard is
yeah, she's the big.
definitely the pioneer who did this.
Yes.
So yeah. Why do you give maybe a history of how you got interested in it, and maybe
Yeah,
a
yeah,
little.
I mean, so when I saw those funny fluctuations in the old bold scanner, because it was taking the TR was like 3 seconds, so I was really always worried about aliasing. I didn't know about it, but somebody told me that, hey, you might be looking at alias signal. This resting state can be all crap, and what are you doing?
Yeah.
I was very worried about that for a long time. And then I went into one of the ISMRMs. It's very good to go to those. For those
Yeah.
youngsters out there, please go. Always meet these masters in person, talk to them, go to their labs, and that's where you got to see things.
Yeah.
Anyway, I went by Pierre Levan's poster, and then I just like Donald Duck walking over the cliff, and then realizing what was that. I was able to go back to his poster, and he had been scanning with a TR of 100 milliseconds. I said, well, yeah, that has to be one slice. He said, no, the whole brain. I went like, huh? How do you do that? And then they had just in the Freiburg, they had, based on Jürgen Henning's talk in ISMRM in Miami OVOC thing, they had developed this optimized spatial time domain where you can gather the whole brain in 10 times per second.
Yeah.
And I went ballistics. I went like, oh God, how much do you want for that sequence? There's nothing. We can bring it to you. I'm like, huh? Hey, and there's a story about Andrei Jesmanovic. I went to Andrei. He was playing in the cellar with the volumenar EPI at the GE 1.5, and he was trying to scan the alpha rhythm. And that was my first. I got to see that it's possible to do that.
Yeah, he was using standard. Well, right, there was echo volume imaging with Alan Song.
Yeah, you know that stuff. Yeah, obviously, yeah.
Yeah, and he actually did have 10 times per or 100 times.
Yeah, exactly. Yeah. I was like, wow. I saw that data. I had a touch on that. I was like, wow, that can be done. But then I said, well, maybe in my lifetime we get this sequence. In the old days, I might be able to see it. But then I walked by Pierre's poster, and they came to us. We just got our first 3 Tesla Skyra, and they installed it.
Really? That's interesting. It was a Siemens. So Henning had worked with Bruker, but he actually had something on a Siemens. The MREG was. That's
it's a Siemens thing. Yes, obviously. Yeah. Freiburg is one of the biggest, if not the biggest, Siemens lab in Germany. So they are very well versed there. So that was Siemens, and we were lucky to get a Siemens, and then we got it working. One wine bottle and some food, and then we went to sauna and dipped on the snow, and they were really happy, and we were also really happy too.
Wow.
That was just it. Very cheap and fantastic. They were helping, like Jakob Aslender and Pierre Levan. Hi guys, thanks a lot. It changed my whole career completely. So I was just worried about aliasing, right? So if you don't sample critically, you might be aliasing basically the cardiorespiratory noise over your ELF signal. Just like, Bob Cox in Milwaukee Resting State Biennale. Remember what he said? Gold signal is mostly crap. And I'm like, Bob, what did you say? Holy cow. And that's what he said. But I think that is related to the non-critical slow sampling, and that's what I've been seeing a lot. So I wanted to see if the low frequency fluctuations were really true that we saw with Vera. And well, obviously the resting state thing is kind of real thing. It's such a huge thing nowadays. But way back then in 99, 97, it wasn't at all. No. And so I was worried, but then we did these scans and took your beautiful paper of the time processing strategies, took the FFT, that's that's where I learned it, it's your paper. And we did the FFT, and we saw these power peaks, right? And the VLF was there, but then the respiration was completely different. Cardiac was even further, and we could separate them by bandpass filtering the data. I was like, and then we can map them
Yeah.
see what's happening on them, and it's like, whoa. And then we see new things like how respiration is modulating the cardiac. We call it
Yes.
CREM.
Yep.
And,
Yep.
and then also, like you were showing already in this fantastic 93 paper, the harmonics of
Yeah.
the fluctuation. So we see the same in cardiac, and we are just publishing a paper soon, hopefully, or submitting it, that also the harmonics are altered in Alzheimer, big time.
Interesting.
Yeah,
So
yeah.
do they go up or down?
Um,
Well, I don't want to put you on the spot, but either.
remember it now. Maybe it was down.
Okay. Well, it makes sense that they go down. Well, if they go down, things go slower, maybe. Like harmonics
Yeah,
are like,
they become more sinusoidal, and
okay.
not like the real heartbeat is like systolic, and then you have ripples. Actually, in MREG, you have a crazy thing because what we thought and Pierre thought that when you look at the cardiac signal, I thought it should grow up, but it's actually going down. When you have the systolic impulse coming, it goes down, and that's what Stefan Bosse showed in the VPI paper, VEPI paper he has on the same issue. So the arterial signal goes down, and it's because of a phase contrast. And so with MREG and with speed, you get two contrasts. You get the bold, and then
Yep.
you have the phase contrasts.
Yes.
But
Yeah.
I've been talking to a lot of physicists, and they kind of don't know much what's going really on with the signals, and it's been it's like I mean,
Yeah.
we see things that are repeating in every living human brain. We see the cardiac resp and all these; they are always there, more or
Yeah.
less, and markedly affected by disease. But then I mean, I don't have enough physics background. I was talking to Olli Grön from Kuopio. He's been a lot in Minnesota,
Yeah.
MRR. And he said, well, it has to be mostly in water. It has to be a phase contrast because that's kind of the only thing you could have there because of the flow. And that's the phase contrast is nothing new. It was shown by Van Schultes and Higgis in 19.
Oh, yeah. Just phase contrast,
It's an
velocity
old,
related phase contrast.
it's an old thing in the arteries with the systolic speed up of phase of the spin. So that's you have two contrast, and then you can start playing with them. Yeah.
Yeah. So let's back up for a second. So one just really quickly, I want to just make sure people understand, like MREG, at least as far as the basic sequence, it's like a stack of spirals. That's
Oh
what
yes. Yeah.
spiral
Exactly.
spanning, and then you kind of go up and down through K-space in a spiral way.
Yeah. So in the classical EPI, you go the K space from the other end side to the other, and that you can fairly quickly scan that the K space with that one too. You can do voluminar EPI, but with MREG, they have these stack of spirals that are going clockwise, and then the next one is going counterclockwise, and then the other one is going again clockwise. So that
Yeah.
cancels out some problems.
Yeah.
They are focusing on the K-space center,
Yep.
and that gives you nice contrast. The voxel size is like a 3 millimeters, so it's not that bad.
Yeah.
You get a brain picture.
Yep.
And the point spread function is not that bad, so it's actually quite optimized thing.
Yeah.
Yeah, so it's like a stack of spirals, like Gary Glover, he has a paper on the
Oh,
spiral.
he loves spiral scan. Yeah,
Yeah.
people like spiral scan. I mean, the problem, I have just two issues with spiral scan. I love spiral scan too. I think it's more efficient, but temporally. But sometimes, like like if you have an off resonance artifact, it causes warping in echoplanar. With spiral, it causes sort of like radial blurring, and so it's hard to correct that. But it's also when you have pulsation, and I especially notice this in visual cortex, when you have pulsation, it causes these rings that kind of go through around the pulsation, like throwing a stone in water, as opposed to with echoplanar, it's just like this line.
Yes. So like from the sagittal sinus, you have this circle around that. Absolutely, we see that too, and I don't know how to. Yeah.
You can look through that. You can learn to look through that, I guess. But I guess the main thing, and this is the problem that a lot of people have, the reconstruction is hard. It's not on a raster. And so you have to somehow register to a raster to do the 2D 4D transform. And that's, you have to do all that offline. And it's like, it's hard to.
Yeah, that's the I think the main shortcoming of the MREG that you have to do it offline, and then you have to do the recon in MATLAB and do the inverse problem solving with the reconstruction program, and it takes time and it takes effort. So you can collect a lot of data fast, but then reconning that into a nifty file takes
That's
a few hours with like few hundred computer cores.
So what's the contrast? So like with, I mean, the TE can't be that long, is it? Or is it inflow? Is it bold? Is it
Well, I mean, the way I see it, I'm not a physicist, take it with a grain of salt. There's the bold response in the slow. I mean, you see all the red networks and activations, and so you see the bold response.
What's the echo time? Is it?
It's like 36 or 34 is sort of the mean.
Okay, that's pretty good.
Yeah. So and I've been asking Pierre that what is the contrast. It is definitely T2 star weighted contrast. So that's what he's saying immediately. So that's it. But because of the speed, you get rid of aliasing and you get a lot of statistical power, and then you get to see new things like CREM and the harmonics and all that kind of stuff, and then you can separate them. And also because of the speed, you can see them moving through the brain. So that's and they do get into very new things like measuring speeds of flow.
Yeah, and you actually, you had this really nice paper recently on spectral analysis of physiologic brain pulsations. It's in human brain mapping in, I guess, 2021. You lay out all the frequencies really nicely as far as that's concerned.
So you can get the vasomotor. We call it the vasomotor frequency.
I want to stop you right there because that's actually a kind of a loaded term in my mind. It's like vasomotor. Because people are looking for vasomotor and they think all of resting is vasomotor.
Yeah. I hear you. Yeah. Well, I knew that this is going to come. Obviously agreed. So, but if you look at Shella Keelholtz and Lucina and Katie Chang's paper, Nature Neuroscience, was it? The big thing there in the variance is the moving thing. So and I mean, if you think of bold activation response, it's actually vasomotor dilation, isn't it? I'm deliberately extending the term here. Sorry about that, but I think it's vasodilation then contraction. And by the way, Mark Nelson popped by here. He had a fantastic explanation. He said that you only need the blood vessel endothelia for that vasodilation. You don't need the astrocytes or neurons. I was like completely blown away by that. If you have he had perfused capillaries and arteries, and in the capillaries he just induced a very small potassium change, and that produced upstream vasodilation, and he had only blood vessel endothelia, nothing else.
Interesting.
I was like, holy cow! Nature Neuroscience. He has a Academy of Science member in US, and wow, just
a potassium change. That's
just potassium. It's automated upstream vasodilation just from potassium via gap junctions. I was like, what? But that's how it is.
Yeah, and the potassium in the brain is produced just by metabolism, or is it sort of like
neuronal activation pushes it out, and then the capillary sense that, and boom.
That's interesting.
I was completely blown away. He gave a fantastic talk, and
interesting. So let me ask you one other question. So when you put things into a power spectrum, So and this is work in my group. There's Josh Faskowitz, and who's been pioneering, and along with Olaf Sporns, the idea of doing what's called edge analysis, and that is where you take time series and you sort of multiply them against each other. It's like a sliding window, but it's with the window width of 1. So you multiply them all together, and so you can kind of look at the moment by moment correlation in time series.
Yes.
And he did this with resting state, and what we're finding, and what they found for the most part, the other papers have found this too, is that bold or resting state fluctuations usually are considered 0.2 hertz or something. And you think, a sine wave. You think a sine wave 0.2 hertz, but in fact, it's like nothing, and then you have a punctate something, and then nothing, and then something. So it's like, and they even got rid of most of their data and just looked at those moments of activation.
Oh yeah,
They
caps.
were able to reconstruct it. So it's not really, people think of a fluctuation like a sine wave, but it's actually, it averages the 0.2 hertz, but it's just all these punctate things, these very short activations. It seems,
Yeah,
there's other ones like you were saying with Shella that might be these slow waves that go over the brain.
yeah. I hear you.
But,
Yeah,
but the
there's
brain
a
connectivity is maybe just driven by the punctate.
well, you were describing that reminds me of Xiao Liu's and Jeff Dean's caps.
Yes, caps.
Co-activation
It's the
pattern.
same thing.
The
Yeah,
same
caps.
thing. Yeah. And that I think they might be more stationary than the propagating ones. And there are things at the very low frequency. I consider the sort of vasomotor part, whatever you have the ball response obviously there for neuronal activation, and then the moving vasomotions, you have them there below zero point 0.1, and 0.1 to 0.34 you have the 0.1, I mean. You have the sort of respiratory range. It varies a lot between people. And then about zero point 0.6 to 5 hertz, you should be looking at the cardiac. This is important because you always have to take the harmonics into account. Just like you were saying, the cardiac is not a sine wave. If you look at the principal cardiac, that's a sine wave, yes. But if you take all the harmonics, it's very edged, and then you have the ripples. That is exactly what you're saying. That's the cardiac. We can see with MREG. Also, respiration is not symmetric. So I hear you. And then again, your early paper shows that with the fold response, it is not a sine wave either.
Right.
It has to sort of go up and then down a little bit and then back. So there may be an undershoot sometimes.
Yeah.
So obviously, So the accuracy also in tandem helps to get these things in a accurate perspective. In addition to the increased statistical power, I'm starting to sound like guy trying to sell something, but.
So that's it's interesting. So I'm surprised that MREG hasn't been distributed more, because it's a whole volume sequence that you can have a TR of 100 milliseconds.
Yes.
And
actually the base space is 80 milliseconds. You could go down to 80, so that would even help more. But they wait for the 20 milliseconds to get to a nice round number. That's what they said. And I think you could put all sorts of stuff to the MREG as well if you knew what you're doing with the Pulsec and others. I mean, like multi-echo, like you were sort of suggesting. Absolutely, definitely.
So here's the thing. So let's switch gears to the glymphatic system itself, because there's a lot of physicists. I mean, physicists love this stuff. I mean, they love thinking about, okay, so you have with the glymphatic system, you have like it's CSF flow, but then it's at all these different rates. It's not in the veins, it's peravascular, and then in the tissue, it's sort of this convective flow, which is faster than diffusion, but slower than flow. And so physicists love to try to figure out sequences like, okay, how can we just look at that? And nobody's quite done it yet.
I think, well, we did have water phantom here, put a hole in a pineapple and showed that we are actually really looking at flow speeds of water with the MREG alone. You don't have to do any other stuff. If you start adding things and gradients and all that kind of control on your sequence, whatever, you start to get slow.
Yep.
And the moment you get slow, you miss cardiac immediately, and then the resp. And then, I mean, it does help to just look at the signal, and then you can, of course, take other, and then you can take 5 minutes, and it's, 6,000 time points. So it's a lot of information already there, and then you can gather like multi-echo phase, whatever. In addition, who prevents you from doing that? So I mean,
but just to reemphasize that, even and this is an important point that everyone should know, is that glymphatic stuff is sort of slow, but the importance of sampling fast really is make sure that that you're seeing those slow frequencies and not alias stuff that's fast.
Oh yes, that's the thing. We have this sort of 1 hertz pump all the time pushing blood into the brain, right? And that's actually not the big pulsator in the brain of human brain. I think the big one is respiration. And so the vasomotor thing, bold response, and the moving vasomotions are in the cortex. The respiration is everywhere, and then the cardiac pulsatility is in the arterial areas, obviously, and in the CSF ventricles.
Yeah.
And the CSF has all of the pulsations, which is kind of weird, but that's what we see every time. Yeah.
So Laura Lewis actually also, she doesn't call it glymphatic system, but she says, well, when a person's asleep and the fourth ventricle, they see this 0.05, so something slower than the typical resting state fluctuations. I'm not sure what to make of that frequency.
I mean, I would definitely call it typical. I mean, so in terms of vasomotor waves, you have two kinds. So 0.1 is like Traube Herring waves, which are thought to be produced by the crosstalk of heart and lung blood flow. So it's a huge volumenar thing, and it's affecting the brain as well. But then the Meyer waves are like 0.03, 0.02 to 0.05. That's, I think, something that Laura might be referring to. And I mean, so whenever you have a change in blood volume, that's affecting the CSF water as well.
Yes.
So it's a complex game, but I think you have to be critically fast. That's, I think, that the main issue. So if you're scanning slowly, then how do you disentangle these effects? It's gonna get tricky. I mean, obviously, it would be nice to have a sub-millimeter voxel ultra. Even, I mean, it should be 30 hertz. Then you would sample the cardiac beat perfectly.
Yeah, and yeah. So the two different strategies, as I see it, right, that people like to try is that going fast, or if they go slow, they try to make, as you said, they try to make their sequence as specific as possible to the specific type of contrast mechanism, like T1, T2, diffusion. Yeah. And so, but you'd like to have them both. You'd like to try to have them both.
Oh yeah. Control what you're seeing in a frequency space. That would be brilliant. Yes.
Yeah. So why don't we back up for a second and just talk about? Could you just describe the glymphatic system and the potential importance of the glymphatic system, and kind of then we can go into talking about the mysteries of it. And
I mean, so I heard Maiken's talk in SFN in Washington 2014, and there was this glymphatic system. I didn't even know what that was. Glial something. I said, well, let's go and listen to it. And then she said that, okay, there are pulsations in the brain driving solutes in the perivascular space as if they were the sewers. And I'm like, wow, that's something. Wow. And she said nicely that the cardiac pulsations in the mice subarachnoid space are the pulsators there. But then she said something really magical that you don't always hear. She said, you have to have something else too. This is not enough. We know this. And by that time, I had my MREG data analyzed, and I saw these three pulsations with Yufeng Zhang's student Xingdi Wang with Shella's QPP algorithms, a beautiful collaboration with all over the planet. Fantastic. So we had these vasomotor pulsation, resp, and cardiac, and we didn't know what they were there for.
Yeah.
And then she just gives me a perfect explanation why we see these propagating waves in the brain all the time.
Yeah.
So I mean, glymphatic is driving solutes in the brain. It's not only waste, it's neurotransmitters, electrolytes, all of that. And it is how the system pushes stuff around. And the waste thing is related, but not everything is understood. We don't know how and where the efflux of the waste, for instance, occurs. We had a really nice defense last Friday here. Steve Pro was here. Abhishek Singh was the student defending the thesis, and we had deep discussions on what's going on, and we really don't know how the efflux is. The thing seems to be that where there are multiple routes of efflux. In animals, the cranial nerves seem to be the big thing. IDES group in Oslo has quite convincingly shown that in humans, at least the nasal olfactory nerve is not a big deal at all.
Okay.
Maybe stuff is pushed immediately through the blood circulation. What we learn from IDES group is that they put contrast media here in the spinal canal, and 2/3 of that contrast gets out of the CSF before it enters the brain. 2/3.
Yeah.
So I think
Wow.
the brain is pushing stuff into the CSF, and I think, to me, intuitively, this explains why we are rotating the CSF all day long, half a liter every day through a tube that is meter long. Why else would we be doing it?
Right.
It's nice
Right.
sewer to throw your things into, and then they get close to liver and
Yep.
and kidneys, where you reuse the proteins again.
Yes.
So, or excrete the things you don't want to. So
Yeah.
I think that's some of the things. And also then, at least in animals, the neck seems to be, this year alone, there's been like 5 Nature Science papers about when you increase the lymphatic vessels here in the neck in animals, you can improve trauma recovery, you can get rid of hydrocephalus.
Yeah.
It's amazing.
Interesting.
Yeah, it's really interesting stuff. Yeah. So and it's connected to the lymphatic system that we have from neck down. But the glymphatic, where does it efflux? That's the question. And the other question is, how does the periarterial water go into the brain? Because aquaporin allows only water. It cannot be the root. It doesn't allow any hydrophilic stuff to go through. But once you are inside the blood-brain barrier, where you actually have the perivascular Virchow-Robin spaces, it's inside the blood-brain barrier. So you might have some small holes between the astrocyte podocytes in the glia limitans, just the outer edge of the perivascular / blood-brain barrier. And there are some suggestions that those gaps can be somehow modulated, but nobody knows how.
Yeah.
So that might be the route where you get stuff in, and
then also not only getting stuff in, but right, waste. I mean, you can imagine there are somewhat large molecules that sort of need to come out, and how do they come out?
Yeah, exactly. People, there's this drawing where you have the artery, and then you push stuff into the perivenous space. Periarterial flow is much better characterized. You can see the CSF tracers going back and forth, but nobody has a picture of perivenous space.
Yeah,
I haven't seen one.
That's interesting.
Except one group, and it's in humans. Kullervo Hynynen's group in Toronto, the FUS people there, Meng and others, they show that when you open blood-brain barrier here in the front, 15 minutes later, you have here in the back of your brain the contrast media that has leaked from the open blood-brain barrier here in the front. It's around a vein in the back of the brain. Yeah. And it doesn't concentrate there with diffusion in 15 minutes. I think that is the first paper to really show that there is convection, not diffusion. Everybody is thinking that it's been diffusion, but it's not. It's not. Right. There's definitely a convection. Yeah. And that's maybe the only paper with perivenous. I might be, of course, wrong. I mean, I haven't seen everything. That's the thing, there's so much thing coming up.
Yeah. Well, let's bring up the issue of how it works well and how it doesn't work well. So it seems like it's activated during sleep.
Yes.
And so the extracellular, I mean, just I was watching one of the talks on this, the extracellular space, doubles in size.
Yes.
And so it seems like the perivascular space, seems like it fluxes in during sleep. And also it seems that it's, if you don't get sleep, obviously you don't get this sort of effect. I don't know what triggers this effect, and high blood pressure, like hypertension, somehow disrupts it. Maybe it disrupts the pressure gradient in which it goes. Who knows? I mean, I don't know exactly. If you could lend insight into maybe, I don't know, maybe just outline what we don't know and what we know in terms of what triggers it with sleep.
Yeah, that's of course the big question, and we don't know much about our own sleep anyway. So that's actually a big diagnostic problem in humans anyway. So we don't see much. If you go to a polysomnography in a hospital, you don't sleep well, do you?
Yeah. Yeah.
You should measure the normal sleep, for to begin with. Yeah, it does increase the clearance increases in sleep. There was a paper showing that should be the other way around, but the one small paper that it actually slows down in deep structures in the brain, but they were using 10 times more contrast than Maiken and others. So they are completely changing
Yeah.
the hydrostatic situation in
Yes.
the animals. That's what I hear. So that might not be a good reference, but in quite a few papers in humans and in animals, it's been shown that if you don't sleep, you get contrast media staying in the brain. Beta amyloid increases immediately after one
Yeah.
night. Nora's and Helene's beautiful paper in class about that. So
Yes.
and of course you feel it. I mean, as a doctor on call, it's horrible after one sleepless night when you've been working a day and a night. It's like horrible. So you really feel it, and you need to go to bed.
Yeah, and I wonder if you could actually even see. I mean, and people have tried to do this, where you try to look at. I mean, have you looked at the pulsations?
Yes. So we have looked at pulsations, and they increase in sleep. First of all, Heta defended her thesis last summer on that. So vasomotor and respiratory pulsations, especially a little bit the cardiac, increase here at the back of the brain, up here, and in the sensory areas. But if you don't sleep and you are sleep deprived, they increase even more. They go crazy. Yeah, pulsations. Yes, it's a marked increase. Yes. So,
but that's if you're sleep deprived and then you sleep, or like
yeah. That was exactly the recovery sleep after. Yeah, you're completely right. Yes. So when you fall asleep after sleep deprivation, they go ballistic. So
Yeah.
we might have tried a few subjects keeping awake, but it was really hard. So
Yeah.
But I think I have a feeling that was also increasing already then. But so the brain is trying to already go into sleep mode, but then you're wake up, and then if you have a disease related to sleep control with narcolepsy. So Matti has this paper on narcoleptic patients, and there you have also you kind of mixed increases and decreases in the pulsations. I think it was vasomotion was increased, and then the card rest was going the other way around. And now we had just now publishing, trying to publish a paper on comparing sleep and awake to narcolepsy. And in some other instances, I don't remember by heart now all of them, but narcolepsy can be even deeper than bigger changes in narcolepsy than awake or sleep. So it goes into a really bizarre state.
Wow.
Brain without hypocretin. So it looks really, really bad in terms of palpations, at least. So yeah.
Is there any way, like, let's say, I mean, this is what people are trying to find, like, I know that, like, in military research or something, they're trying to figure out how they can keep a brain working well without sleep. And so the trick would be to somehow activate the glymphatic system
While
when
awake.
you're awake.
Yeah. Well, I mean, but I think the sleep means that you have the extracellular place open and you have flushing there, and your potassium levels go down. And like Maiken has in science paper shown that quite a few of the electrolytes go down.
Okay.
And then you have loss of neuronal activity, potassium going down, calcium, and neurons don't work. So and
even in the context of lack of sleep, even if people, like, for instance, let's say the glymphatic system people find is the reason, a big reason why people get Alzheimer's. And is there any way of enhancing, just saying sleep more may not help, but is there are people working on ways of, like, understanding the mechanism by which the glymphatic system is triggered by sleep?
So I'm a PI in a EU JBND consortium. Maiken is also there. We are trying to figure out ways to increase the glymphatic efflux. And at least, I mean, we have a paper on this January, Jari Jukkala's paper, where we were able to increase the glymphatic efflux like 4 times into the blood via the lymphatic, but that was animals. We have done phase 1 on humans, and it's looking good.
So what do you do?
We do a little bit of electroacoustic stimulation and then some drugs to the same way we had in the animals to do some lower blood pressure, and that induces then increased pressure intracranially, vasodilation, and then we can see nice things coming up. So we might be able to tease up that, but those are healthy controls. Then let's see what happens in patients, because I'm a little bit pessimistic. There are 200 studies in Alzheimer, and everything is showing that there's no cure till now. So let's see what happens. We are trying to get into phase 2 with that. So, and of course, the other option would be to try to clean the brain with the focused ultrasound.
Yeah. I was going to mention that you had talked about that. I saw a talk that you gave in which you, even with people with traumatic brain injury, doing focused ultrasound to open up the blood-brain barrier seems to help survival. I mean, is that right?
Yeah. I mean, the focused ultrasound has been studied in humans and in animals, and animals have been shown to have a recovery of memory. Mice models, but of course mice don't produce amyloid, and that's foreign to them, and then it's such a small brain that you can maybe clear that more easily. It's been tried from small areas in humans and hasn't worked. The Alzheimer trajectory was the same as in ADNI data. So they had 26 in Kullervo's group in Toronto. They had 26 patients treated 6 times, hippocampal and default mode areas, small areas, not the whole brain. Kind of a safety study, but unfortunately no effect. I think you should clear the whole brain, and that's kind of it. That's what we do in brain lymphoma patients, and we can, with mannitol blood-brain barrier open, that's another story, but we can cure brain lymphoma there. So yeah.
Wow.
But yeah, the focused ultrasound opening of the blood-brain barrier actually increases the glymphatic clearance. The sewers get stuffed with like albumin that has been injected into the brain. It increases the glymphatic clearance for sure, yes. For the trauma paper you were referring to, that was stuff that they were doing basically to increase the lymphatic pulsation.
Okay.
So they have a trauma, and that trauma induces pain and sympathetic drive that sort of stops the lymphatic neck pulsations. And if you return that with one adrenergic inhibition with PPA drugs, a cocktail of 3 adrenergic blockers, they reinstall the lymphatic pulsations, and they literally can get chunks of garbage out of the brain. So if you stop the adrenergic sympathostimulus and release the pulsations, then you can, and the mice they recuperate better for a longer period. So they get much better cognitive wise as well. The edema is minimized, and then they get much better in the long run. Yeah. So there are ways to do it, and I mean, sleep isn't the only thing. Maybe we can increase the glymphatic efflux by drugs or other ways, but we are not there yet. So we need to work and do more science. Yeah.
Or have a clear way of measuring it. So, just related to that a little bit. I was actually wondering about edema, for instance, with when you have traumatic brain injury, the cells swell, and is that at all related to? I mean, do people know the mechanism for edema? I mean, I guess the reason why.
Funny, you should ask a brilliant question, because Maiken got last year. She got in Oslo. She got the Anders Jahre Prize, which is a huge honor, and I was lucky to participate there. And she asked the same question: in brain trauma, what do you think is happening? Is it good or bad when you have edema in the brain after trauma? And nobody said anything. Well, I said, well, somebody has to say something. I was stupid enough to raise my hand. I said, well, maybe you have, edema produced by the cells there, and it's a natural thing, and it's helping to cure the brain and blah, blah, get stuff out of the brain. I didn't know.
Yeah.
She said, no, it's actually an abnormal situation. Quite a big part of it is caused by the blockage of the lymphatic system here in the neck. We are looking maybe sometimes in the wrong spot. That's what she showed, at least in the mice. When you increase the pulsation here, the edema is not completely gone, because obviously you have a trauma there and there's an inflammatory reaction to the debris and all that. But she has those videos in her talk, and in that video where you have the trauma and then the blocking panadrenergic inhibition, you see chunks of stuff getting out of the brain, and they were saying that those might well be chunks of cells. So the efflux system has to be quite big and open, and that's what you see in the lymphatic vessels here. It's amazing stuff. So it's not only the brain, but also the whole body that you need to start looking. So that's what we always say, that okay, you have to be holistic, but you should know what to do and what to look specifically, though. So
that's really interesting. Yeah, I mean, it seems that in general, our initially looking at the glymphatic system and then other systems, it seems that the focus on brain health by understanding the waste clearance systems seems like that's growing, and that seems like it's sort of been neglected in that regard.
We have been looking at least half a dozen diseases, and every single one of them has at least one of these pulsations I describe here badly wrong. In the old days, I mean, with fMRI, I could find a 3 or 4 voxels difference. Nowadays, I'm quite disappointed if I don't get a result covering half of the brain.
Wow.
So yeah, exactly that. And I mean, it's a complete game changer. I won't be able to go back to slow scanning because of these results. It's just the separation of these phenomena gives you so much precision that it's just unbelievable, and gives you individual level diagnostics. We can predict brain lymphoma mortality 100% from scans, MREG scans taken before the treatment. We do open the blood-brain barrier 6 times. That's the only way to cure them. Otherwise, they are dead.
Yeah.
But we have this treatment with mannitol, intraarterial, and chemo, and we can cure them. Just looking at how much the signal variation is, standard deviation divided by mean. Very simple thing. And you can get 100% accuracy. Another stuff you can do in Alzheimer using ADNI data, by the way, not our data, it was ADNI data.
Yeah.
Timo Tuovinen found out that if you look at the CV, standard deviation divided by the mean of the bold signal voxel by voxel, you can see that the CV is changing permanently half a year before the cognition collapses.
Wow. So if you do just the standard deviation versus the mean, then you don't have to sample that fast if you're not trying to get the standard deviation.
Yes, you're right. That's a perfect. Thanks. That's a very good point, because then you don't have to necessarily always have the ultra fast. You can, if you scan for a few minutes, you can sort of get the variance. You get the idea in a way.
Yeah.
Exactly what you said. We had normal scanners and then MREG as well. All of them showed the same thing. 3 patient cohorts. So.
But it might be interesting to look at the standard deviation of specific frequencies, and you might get even stronger.
Yeah. That's what we're doing. Yeah, I love the FFT thing, so we are doing that. But amplitude is very close to standard deviation, right? So it's just the same thing basically. FFT power or amplitude analysis, and then that's what we've been doing. Yeah, absolutely, yes. Once you get into the bands, it's amazing. It's amazing. Yeah. I won't be able to go back to old bold. No way. I mean, if somebody comes to me and say, "Let's do it," but your point is good in a way that you still can do it with the CV. You can do stuff also with the slow. I call it slow bold, like Eric Clapton, slow hand. It's good. Jim Hendrix is better in other things, but we'll go there.
But yeah, that's actually sort of does bring up the point of how research is related to vendors. I mean, vendors could probably care less about MREG because they have their cardiac imaging, they have their other stuff. It's not really, they don't see a clinical market for fMRI, and so they're not going to develop this. And so it's a little bit of a chicken and egg problem of like, how do you get a sequence like MREG, such that it creates clinical market such that vendors then are interested in developing MREG. And it's
you touched the core point, and how are we going to get the recon in a way much faster that it would be feasible? That's a thing. I mean, I have a feeling that this should be doable in a much faster way. The recon, that's the thing.
yeah.
For instance, the brain lymphoma. The colleagues here said there's no way you can predict it from any anatomical image, laboratory, clinical biomarkers, anything other than MREG.
Wow.
No way. And it's like 100% accuracy. It's Walter Poltojanen's paper in, I think it's HPM 22. So I mean, to me, it has, I mean, I'm a neuroradiologist. I want to do fMRI in the clinics. I hate the black and white and gray, 50 shades of gray in the anatomical images. It's like, I don't hate them, but I want the colors.
Yeah.
And with fMRI, you got the colors there, and it took only a quarter of a century to get there. But I mean, it has to have a huge added value, just exactly what you said, that the vendors would take. Of course, it's money and effort, and it's not easy. Yeah, absolutely. So
So let's say this is a hugely unique. So it sounds like with MREG you have at least something that's uniquely clinically useful.
Oh yeah,
you're using it. So how would that become a standard clinical practice or something on the scanners? I mean, there's a lot of hurdles to clear from you doing it on your patients and saying, oh, this predicts 100% versus the world doing it on a daily basis. How does that happen?
Yeah. Well, you asking me? Oh God.
Well, I mean, I thought about this a lot. Like, certainly, you have to have like FDA approval and
and how to do the recon, how do you do analysis? Well, in a way, it becomes easier when you can separate the phenomena and focus on what you want. I mean, we have to produce something really valuable, like the prediction of mortality or something like that.
Yes. Yeah. Something clear.
Something clear that you cannot do by any other means. That's obviously the thing. One of the things I have been sort of trying to do is to develop these maps of glymphatic failure, where the glymphatic slows down. Like in brain lymphoma, we just saw the optical flow results that the lymphoma cells usually grow around in the perivascular spaces, like in the lymphatic vessels they do.
Yeah.
They stuff it around, and they prevent the glymphatic solute transport there. But once we get it cleared, we just got a nice result that this problem goes away. Once we get rid of the disease, it nearly normalizes the speed of the movement of the pulse. So it seems that we are really looking at somehow the glymphatic clearance and the speeds and everything we see. It's somehow related to the speed of CSF movement, but I don't know how to change the world.
Well, it seems that you need like a third-party vendor that somehow, it seems that, and it's interesting because it can't necessarily be like PulseSec is this third-party pulse sequence thing. I mean, and also you have open access processing, but it seems like you need a vendor that cares about making money, unfortunately, that takes responsibility to push it to FDA clearance, because it's very expensive to do that.
Oh yeah.
And you have to have clinical trials. It's expensive to get grants. And then finally, I don't know where that step is made where the vendors might say, hey, this is a potential clinical market. Let's do this. And then they put all this work into it. And maybe that's how it's done. Then then it slowly builds that way. But it seems like you have to like kind of get the attention to the big vendors and maybe a third party vendor, and then sort of work out all the nuts and bolts of and even processing. Like, if you just have a technologist pushing a button and a radiologist looking at it, what's going to happen?
And interpreting it that okay, these pulsations look like this. What is the disease? Yeah, you're completely right. That's a good question. I don't have the answers, and I mean, first of all, it requires people in the scientific community to get interested and involved. I think one thing is to get normalized data baseline. I've been dreaming of this GMREG network in Finland, but even better nationally, internationally, everywhere. That's even better. Collecting data into the same repository. I think one of the problems in MR is that we don't have a SI unit, and every scanner is kind of different a little bit, vendors
Yeah.
especially, and
Yeah.
how to calibrate. But I was thinking that if we do the optical flow imaging, it's meters per second, right? And that's SI units. And
Yeah.
once you're there, then it actually gives you
Interesting. Yeah.
calibration in a way, right?
Yeah. Yeah.
So I mean, there's a lot of work to be done, but once you get into flow speeds, then you get into these nice equations that you can start doing things that you haven't been able to do so far.
Yes.
And I mean, just like I love Steve's and the biobank, they are collecting the 100,000 data set, which is like, oh god. And so something similar, maybe in a smaller scale, but a few thousand subjects over the whole lifespan, and then you can start to see how the flows go,
Yeah.
all of the three pulsations. Why I am so interested in is that basically every disease we have touched shows something really, really wrong there. So that's
Interesting.
my motivation so far. But your question is, of course, very important. How to go over the FDA, CE,
Well, I mean,
and show that.
yeah, I think in general, vendors could do probably a better job sort of setting up a platform that allows people to test potentially clinically useful things that would create markets. I mean, this is another conversation
Oh
altogether.
yeah, I hear you. Oh, big time. Yes, yes, obviously. But
Because right now, yeah, we're in the dark of hacking away, getting MREG working. Maybe one or two centers have it, and
yeah, exactly. Yes, and it's not enough, and we should definitely like collaborate and get data together in a repository. And I mean, we could give a few hundred brains already scanned. So
yeah.
honestly,
Well, that would be interesting.
the GDPR problem is, of course, a hurdle again, which is not helping us at all. And all these scientific regulatory things are getting really hard everywhere, at least in Europe.
Yeah, like in terms of sharing databases,
Yes,
the
that one,
privacy.
and then also that MREG is not a CE marks sequence, so that like you were saying, FDA approval is missing. So at least in Finland, it requires a national FIMEA approval. We have that, of course, but it's slowing things down a bit.
Yeah. Yeah.
And yeah, indeed, it would be nice to get it. Yeah, I think we need to get data and the recon problem solved somehow. So if somebody is listening with the bright mind and how to get inverse imaging fast in a nice, doable way,
That's
let
interesting.
go. Yeah.
You would think a machine learning algorithm could,
I
there's been
mean,
made so much
I
progress.
agree. I mean, some sort of new way of machine learning that can really sensitively look the time point to time point variations, like the cardiac and all that happening, and not producing a straight line like so far has been going on. So like plastic images. I think it exists in the future. Somebody
Yep.
comes up with an idea. I mean, what I love in ISMRM, I mean, since '99, I've been going there, and I said, well, now I've seen all. It cannot be anything else now. And then you go to the lectures and you see, holy cow, what have they been doing? It's just every time new and new and brighter and more beautiful things. It's amazing. So
Yeah.
I have big faith in that. I hope we can get it rolling and in the future have more money on science than war, if you know what I mean.
Yeah.
So,
Yeah. No, I think you're right. ISMRM, I mean, it's interesting comparing OHBM with ISMRM. OHBM, and they both have their strong points, but I like to go to both because
absolutely. Yes.
OHBM has like the latest in, sort of processing methods, big data,
Yes.
understanding the brain. But then ISMRM, it's like with the MR sequences and with the recon with that, and maybe contrast mechanisms.
Yes.
They're like several years beyond typically what's presented at OHBM.
Yeah, it takes a while to get into the HBM, but yeah, but both are needed. I hear you absolutely. That's obvious, true. Yeah.
Yeah. So as far as what you're working on right now, uh, and yeah, we have like I mentioned to you before I talked to you, so I have a postdoc, Barak Aiken, has worked with Hennig, and he has the MREG sequence, and it works on one of our scanners a little bit. Yeah, it's actually a neurovocal scanner. It's not ours. We use hers, and
Wow.
and she actually is very interested in this as well. So we might be collaborating with her with MREG potentially. That's exciting. So what do you think is what's exciting to you right now? Aside from, I mean, obviously all of this is exciting, but what are you like? And you sort of hinted at some of your main questions, like applied towards clinical applications. But are there any mechanisms or other things that you think are able to be pulled out using MREG in terms of?
I think MREG is a way to collect case space, and you can do more, obviously, and much more than what you could do then with multiple echoes and phases and diffusion gradients, Tay-Scaled dynamics and whatever out there. You could do that. I've been looking at just normal variation. I've been doing a lot of stuff with flip angles, and already that produces quite a bit of difference. I would have wanted to talk to Jerzy, but yeah, it's not possible anymore. So
No.
that's the beautiful flip angle paper you and Jerzy and others.
Yeah.
So that's already something, and there's a huge change. Yes.
Well, that's interesting with the flip angles. So let's just talk about that for a second. I mean, right, if you vary flip angle, everyone thinks that with bold you should scan at the Ernst angle, but because that's sort of what optimizes the signal to noise based on the T1. But in fact, because physiologic noise dominates, you can go to flip angles down to 10 degrees and still not lose any temporal signal to noise.
Well, so we have been using flip angle 5 in quite a few of our published papers, and it works like a charm. It gives, for instance, the cardiac pulse is much more clearly different from the other noise. We have been trying to publish results on that a lot, but we don't kind of have the physical understanding of what the hell is going on in the spin gymnastics.
Let's talk to you more about that.
The history, spin history effects and all that. I've been reading, and others have been reading the Castillo Gonzalez paper where Jerzy is the last one. So it's well, again, MCW stuff.
Yeah, it's MCW sort of like things. So so if you lower the flip angle, you reduce the amount of magnetization that's saturated versus unsaturated. And so if you have inflow from the cardiac, that might be a little bit, it becomes saturated a little bit quicker. So if you have the reduced flip angle, it doesn't like stick around a long time. Once it's flowed in, and that's maybe why you might get a cleaner signal, because you don't have mixing of one heartbeat to the next.
Oh God, yes. I need to write that down. Honestly, I was wanting to talk to Jerzy about this like over a beer or two. What is going on, because it's such a different ball game. Flip angle 5 and 25, and then there's we should have done flip angle 10, 15, 20, blah blah, and go. It changes the ball game. So this is an obvious thing to do immediately, and since you know the thing much better than we do, please go ahead.
We'll try.
And Pulsec in collaboration with Maxim's people in Freiburg, you can maybe start adding things in the Pulsec and playing around with that. So that might change things quite a bit.
Interesting.
The normal variance, how much the pulsation change. We have a few hundred controls now. And it's amazing when I have really hard time quite believing it myself, because when you take a control person and compare it to control, they don't change in the same age and sex. But over age, it does change. But when you compare it to a pathology, it's just crazy how big the changes are. It's a complete game changer for pathology contrast for imaging in fMRI.
That is so interesting.
It is. I mean, it is so weird and so strong that it's unbelievable almost sometimes.
Yeah, what's really interesting is that, in the reason why maybe it was a blind spot in standard clinical imaging is because everyone is trying to get rid of all the pulsations. They're trying to look through them.
Oh yeah, the stone that the builders forsake becomes the cornerstone.
Yeah.
yeah. Oh, I mean, what's in the noise? Weisskopf and Burdo paper way back. Yes, I mean, I have really hard time nowadays with the term noise because it's well, thermal noise is thermal noise,
Yeah.
yes, and it's random and pink and whatever. But if you take the MREG data and produce voxel by voxel the FFT spectrum like you did here in your paper, and then add them up together, you can see that the signal to noise can be like in a frequency domain way compared to the noise level it can be 200
Yes.
or even much more. It's just unbelievable. It is we don't have to measure the heart rate nor the respiration because we see it from the MREG data. We have several papers showing that it's one-to-one correlation,
That's
absolutely precise.
actually. So
So
what's really interesting? Yeah, what's really interesting that you just mentioned that because it's like everyone says, "Oh, the physiologic noise limits your temporal signal noise to about 100 to 1." But if what you're interested in
yes,
is the physiologic noise, then
yeah,
there's no
yeah.
limit. There's no limit in what the signal must be.
It is, and let me tell you, pathology, it's there. I mean, almost every time there's the pulsations are increase in power in basically every disease, almost not every time, but quite a few. It's increasing, even though the people are breathing and pumping their blood in the same way. How in the hell does that happen in the brain? I don't know. That's where I think we need to get people involved. And Athena Demerci just, we are having a discussion tomorrow. She wants to get the MREC stuff rolling. So, and you having that's, that's a big sign. I don't care about the storm anymore. That's excellent. Yes.
Oh,
No,
we're going to try to work out. Yeah.
we have to produce this kind of uh, data pool where we can, because of the recon is so difficult, we just put data together and controls and have gender, age there, and then I recommend strongly blood pressure,
Yes.
EMI you have from the scanner and so on. All of them affected, all of them. Let me tell you, all of them, and it
I
is.
even have a preliminary study where I'm trying to look at. We have blood pressure monitored during the scan, but also like just correlating baseline blood pressure with anything like pulsation magnitudes and yeah, or even bold response or
Yeah,
like
yeah, 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 yeah. Yeah,
so interesting.
absolutely. It's a basic thing, and it's funny. It's not been done.
No,
That's
no,
I don't realize. I mean, we started from the beginning. We have a few hundred cases and some patients too. We measure it all the time, every time. Yes.
yeah. And so what is? I mean, it's funny because blood pressure is one of the only predictive measures that's so powerful
Yeah.
that if you're above some level of hypertension, suddenly all your mortality and everything just goes through the roof.
Yeah,
And
yeah,
so it
exactly.
seems to make sense to look at that relationship.
Oh
Yeah.
yes, sir. The guy who wrote the amplitude paper you mentioned, 21. His third paper in thesis is about blood pressure and
Oh,
yeah.
really? I'll
So
have to ask you about that.
yeah,
But
yeah, yeah.
yeah.
Well, you know everything. So we definitely need to talk and see what goes on and somehow get this, GMREC network, Glymphatic MREG, the GMREC network. I don't know what we call it, but and I don't know where to put the data because there's so ridiculous amounts of data from one brain. It's 6,000 volumes. It's like, but
Wow.
luckily nowadays we have big disks, so where we can put it. Yeah.
Yeah. Well, maybe we could start a database.
Yeah,
Um,
yeah, something like that. We need to, and it doesn't cost much, and if you put data in and have like barats towards the 1,000 connectome stuff,
yep.
where you put the data, it really makes things faster. And yeah, yeah.
Yeah. I mean,
I,
there's so much use that comes from data pooling and data sharing. That
oh yeah, yeah. People don't have to reinvent the
right.
wheel all the time.
Yeah.
Yeah,
Yeah. And there's so many different ways, and there's so many different ways of looking at the data and
oh
processing.
yeah. I mean, we we, we have just been using FFT, CV, and spectral entropy, and then the optical flow, and that's it. And
Yeah.
every time we find something like half of the brain.
That's
Well,
so
you know how much. I mean, it's FSL. I'm sorry, randomize or Lipsia corrected for, and we usually increase from zero point zero 0.05 significance to 0.001 or more, or I mean lower. because sometimes it's just the whole brain and nobody believes us.
Yeah. Yeah.
It's a complete game changer. Yeah.
So interesting.
Yeah.
So, end the podcast on sort of the thought of, like, for instance, what advice that you would give, even like young researchers or even professors who are maybe even thinking of getting into this. I mean, what advice would you give to people?
Well, I think you should read the old literature, and then, like I said, talk to people like you and maybe me too, and see the old experience, and then collaborate with people and interact. I think that's the quickest way around,
Yeah.
and do bold things, right?
Yeah.
You know.
Just try things.
Yeah,
Yes.
exactly. Yeah, and you go into territories like I remember Elias Zerhouni said in some meetings that I call it Zerhouni mapping, because you should be looking not in the places where others are looking and what is known, but look at the blind spots. I
That's
think
exactly.
that's where the money is, and don't be afraid. Just go it and try it out. I mean, of course, things have to be real.
Yep.
Yeah,
Yep.
but science is good in a way that if you don't have a small p-value, you don't have a effect.
Yeah. And
Quite
right.
you're quite humble.
Yeah.
Yeah.
And I think actually you're right. I think a lot of science is slowed down by dogma. Like, for instance, if you're asked most people who do fMRI, like, oh yeah, there's the cardiac and respiratory fluctuations. That's it. And that's interesting. It's not that interesting, but we try to get rid of it. But that's not the story.
That's where the money is, actually.
Right.
The blood does produce the energy for the brain, and the respiration is getting the venous blood out of the brain. And if those are changed, bye-bye. It's immediately something goes wrong. Yeah.
So that just made me think of one more question I have to ask about.
Yeah.
So there's work from Arno Willinger's group at Max Planck and also Blaise Frederick. And they look at this sort of latency of, like, they look at these sort of pressure waves that sort of, like, from resting state, and they can actually predict, where they're looking at bold, where flow deficits are and things like that. But I wonder if, using MREG and trying to look at different onsets of different frequencies that might be interesting in terms of, looking at the wave of glymphatic, flow or things like that. That who knows?
Yeah, I mean, I know Blaise he's been doing TR 100 works and these, and he's been looking at the multiplexed slab stuff where we are collecting the brain fast but in slice ways, and trying to extrapolate then the pulsation in the brain. So I know that data. I mean, already in the VLF range below 0.1 hertz, from 0 to 0.1, there are quite a few things going on. The Traubenherring, the Meyerian, and then we've just pumped into something. The CSF is doing something all of its own. I mean, it's not the same as in the brain, and there are so many things. I mean, that's why I'm trying to say to people, hey, come on, this is the next big beef. To be honest, I think in my point of view, the task activation and functional resting state connectivity, they are at the bone level. The meat is gone, but the nice Texas juicy steak is now physiological, if you know what I mean. Take a beer and enjoy, and there's a lot of meat for everybody here. So that's, I think, let's do it together. I think the big issue is that we have the diseases to fight, not between each others. The more you collaborate, the stronger you are, the faster you get to results, and the diseases are where we should be fighting and not each other. Goes into many realms of life, So
it's not a zero-sum game. It's sort of like everyone wins when we collaborate.
Yeah, absolutely, yeah.
All right. Well, on that level, I mean, there's so much more we could talk about, but at the same time,
we could go all along.
Yeah. No, it's thank you. I mean, obviously I've known you for a long time. You've helped me out just recently when I was invited. I'd have to mention it. I was invited to give to be. It's funny. I got this email and I didn't even know what this was. Is to be an opponent in a thesis
Yes.
defense. I thought, well, I've been part of a committee. That could be, but no, they're in Northern Europe, in Finland especially. You have one person, and they're the opponent for the person defending their thesis. And obviously, you have more experience in this and giving on what to wear, what to.
Yeah, I've done it 15 times, once myself, and then 14 my students.
Oh my gosh!
So yeah, we have one opponent. We have the guys wear tailcoats and sort of it's quite formal. It's an old German tradition we have. We have 2 reviewers before the thesis, and we go through and correct it. But then at the day of the thesis defense, we go. It's a public defense, and yeah, like you had the tailcoat, right? Or you had the dark suit. Well, the same here. Sometimes we have the black suit, sometimes the tailcoat, but the tailcoat is the old edition. Yes.
You have like a top hat or something like that.
Oh
as well?
Yeah, we have the hat. We have a sword to defend the truth.
Oh my gosh!
Yeah,
we
have all these old things. So yes, it's
have a wonderful tradition.
Yeah. I think it sort of lifts the level of appreciation for the science in a new level. I think it's requires a little bit extra work, but it's a wonderful day. It's just one of those days you have in your life. It's unforgettable. It's perfect. And you went to Helsinki for that, and that was really nice. And you had a nice group and defense. That's what I hear. So
the defense went well. Everything was great. We're even collaborating with them on a few other things with Lara from RISE. So.
Well, yeah, Lauri has great stuff. Yes, I know him very well. So yeah. Excellent group.
Yeah. So anyway, that was fun, and thanks again. I look forward to hopefully seeing you at the future OHBM and ISMRM.
Yeah, surely coming over to ISMRM hopefully, and see you there at least. And thanks for the opportunity to talk. This is very important, and to bring out the gospel of MREG.
well, thank you. Hopefully, it's being maybe it will be spread a little bit more today. So
yeah. Okay.
All right. Thanks. Thank you.
Okay.
Bye.
Bye.
Neurosalience is brought to you by Organization for Human Brain Mapping. This week's episode is produced by Xiqian Michel Li and Ömer Faruk Gülban.